Well, thank you for the warm welcome. It's always a pleasure to come back to a place that you've spent many years and have many fond memories and walk the hallways and see many friendly, familiar faces, but also all the great changes that are occurring within the school. And I appreciate the snow to make me feel welcome at Colorado, but also the warm environment here. And what I'd like to do is tell you a little bit about some of the research that is ongoing at my lab, where we're very interested in designing different types of biomaterials, soft biomaterials, polymers, and how you can use them in different ways to answer biological questions. And I'll give some motivation of how we started in this and why we're interested in such types of questions. So one relates to the growing use in medicine of using cells to get our body to heal itself. Now some of these ideas aren't very new. So some of you may have had blood transfusions, and they've been around for many decades and saved lots of lives. Bone marrow transplants to treat certain types of cancers. Islet transplantations for patients that have very severe diabetes that can't be regulated by insulin injections. There are now transplants of islets from donors. An area that I've worked in with chondrocytes, so those are cells that can form cartilage tissue, and strategies to get cartilage to repair itself rather than having synthetic implants or replacement joints. And in fact, if you go to the FDA's website and you do a little search on all the clinical trials that are going on and using cells for different therapies, you'll get 20,000 or more hits just using stem cells in those therapies. So not things like blood transfusions. And they're injecting cells into patients' hearts after they've had a heart attack to try to get heart muscle to grow. People with Parkinson's disease, different types of neuronal and neural precursor cells being used. Even the use of human-derived embryonic stem cells differentiated to oligodendrocytes are being used as a therapy to treat spinal cord injuries. So this is growing and it's becoming more prominent and interesting. But some of the motivation and the types of questions that we ask is that, so as these cell-based therapies are growing, one of the problems or the issues is that many of these treatments are suboptimal. And I say with low cell engraftment. So that means when you inject the cells, only 1 or 2% of them survive. 98% or more oftentimes don't survive. And so part of that reason, we believe, might be because cells in our body, and many of the cells in our body, reside in a tissue, in a material niche. And when you just deliver cells, that's only one half of the question. And the other is, as we start to use stem cells and different types of cells that can become multiple different cell types in our bodies, right now, many of the techniques that we use to culture these cells in vitro rely on using things like polystyrene, things that we as engineers might use as an engineering plastic because of its good optical properties. And cells are cultured on these types of environments, and many times we lose some information because this context isn't so biologically relevant. So how can materials help with this? And so I want to give a little bit of perspective then. So what I'd like to tell you about today is, so we're really interested. So if this is a cell and it's interacting with this material environment, binding with certain receptors on its surface to cues out here, talking to another cell, binding different types of soluble proteins or growth factors to get instructions. And we've been very interested from an applied side of making these material environments to regrow and regenerate tissue structures. So we worked with cartilage, getting our bones to heal faster, different heart valves, especially for babies that are born with heart valve defects, some islet transplants. But what I'd like to say about this is that while we've made great strides in many of these tissue types, and the field has in general, that one of our failings is that if I pick something like cartilage, we can regenerate cartilage, but an aspect of its function that's main elusive is that every time we regrow cartilage, its mechanical properties are inferior to the native cartilage tissue that's found in your joints. Well, why is that? We still don't know that very well, and it gets down to this question of trying to understand how cells receive information from this environment. And if I could better understand how cells translate a signal from a material, then maybe I could design better types of biomaterial niches to grow these tissues. And that's what I'd like to tell you about today, is some of our recent efforts in designing new types of polymer materials where we use light to tune material properties, and we use different types of orthogonal reactions so we can look at multiple signals or cues. And then if we do that, then perhaps we can design experiments such that we can understand how can we promote a cell to move or migrate in a certain direction? How can we cause it to differentiate into a certain cell type? Or understand how it senses a mechanical signal from its environment? Or perhaps even influence how its secretory properties. So what I mean by that is how fast it makes and elaborates something like a tissue. All right, so here's one more set of an example of why I think material environments can help us. So these are cells found in your heart valves. They're valvular interstitial cells, VICs. And if I'm a biologist trying to study and understand these cells inside your heart valve, what I would do is I would digest away the heart valve tissue and then culture those cells on a flat plate, this tissue culture polystyrene. And this is what the cells would look like. That's their nucleus, and they interact with that matrix, and they form these long stress fibers. But let me tell you something about these cells. So if you were to look at the molecular level details of all the things those cells are doing, the genes that they're turning on and off, and I don't care too much about the details here, but this is a gene array, a chip, where I can look at all the genes that the cells are turning up and down. And over here, this is what happens when you plate these cells. And you can kind of see this pattern of expression and how the cells all cluster together and behave similarly when they plate. But if you weren't even trained in this field at all, here's what the cells look like right once you isolate them from the tissue, before you plate them at all. And the pattern of expressions of genes are totally different. So here's one basic motivation, is that the instant you take these cells out of the tissue and just put them on these plates, they're behaving differently. And what you'd like to have is a way to culture these cells so it behaves more like it is in the tissue. And so that's what I'd like to tell you about today, is how we can use hydrogel systems to culture cells in a more physiologically relevant context, and use new types of chemistries to answer questions about how cells sitting inside of this hydrogel will get information, so I can learn which signals to put inside of that hydrogel. And so hydrogels, if you could look at the molecular level details of this. So hydrogels are simply macromolecules that interact with one another, and these interactions prevent those macromolecules from dissolving. So they're hydrophilic molecules, and that causes them to imbibe large amounts of water, but the interactions between the chains prevent dissolution. And so that's very desirable for culturing many different cell types in and on, because the high water content allows for facile transport of diffusion, it allows cells to signal to one another, and the high water content and elasticity also matches many properties of many of our tissues in our body, our soft tissues. So why is an engineer interested in these types of questions? Well, because understanding and manipulating this cell environment requires knowledge of understanding things on multiple timescales. What happens the minute I put cells inside of here, to the several days as it begins to remodel this environment, to several weeks as I want to coordinate complex systems of tissue regeneration, as well as on multiple size scales, from the size of that receptor on the cell to tens of microns of two cells migrating together to millimeters, the size of tissues. And so that's where a lot of engineering analysis and principles can come into play as we look at scaling up. So I'm going to tell you about one particular type of hydrogel system. So it's based on polyethylene glycol, so PEG-based hydrogels. And one of the reasons that we like PEG-based hydrogels is when you put cells inside of this environment, PEG imbibes large amounts of water, and it also prevents a lot of non-specific interactions that occur when you culture cells. So in a cell culture milieu, you have lots of different proteins in the media, and if you have many materials foul with those proteins, and it provides a background signal. So PEG provides as close to what you might call a blank slate for culturing cells, and that's going to be important because I want to introduce cues in a systematic way to see how cells respond to that. Now, the simplest PEG chemistry that we began working on 15 years ago, you just take this polyethylene glycol, and you put on acrylate or methacrylate groups or some type of vinyl moiety that when I induce a chain polymerization, I can take these linear molecules and cross-link them to form these water-swollen networks. And the conditions are such that I can identify these. So this can all occur in the presence of cells. So I can just add this to a cell culture suspension and cause gelation to form and entrap the gels. Now, since I know that Purdue has recently celebrated its Centennial, and it was very much a fun part of my history to be an undergrad here, I threw in one page from my undergrad laboratory notebook. So when I was an undergrad student here, I was fortunate to not only have many great instructors, but also to get involved in the labs. And so I worked in Nicholas Peppis's lab, and at the time I was also taking kinetics from Nick Delgas and learning all about kinetic processes. And so here, that PEG molecule with the acrylates, Nicholas was trying to explain to me the initiation and propagation events. And I even, if you probably can't see in the back, but this is January 17th, 1991. My interest in these types of chemistries goes way back even to my time here at Purdue. But I was very thankful for that experience, and I know many of you as graduate students also mentor undergraduates, and I think it's a wonderful thing to do. Okay, so these PEG-based matrices, let's get back to our story then. So the thing about these PEG-based chemistries is that they're very nice for culturing cells in 3 dimensions and encapsulating them, but because cells don't recognize the PEG chemistry and it doesn't have signals, most cells take on this spherical morphology. And so these are the cartilage cells, the chondrocytes. And in cartilage, they're naturally spherical. So they can be very great chemistries for culturing these types of cells. These are cells found in your pancreas, the islets, and they grow into big cell clusters. So it's also very nice for culturing cells where you want to promote cell-cell interactions and grow large spherical cell aggregates. But the problem is that many, many cells want to interact with the surrounding matrix and get signals, and they also want to migrate through this tissue. And so if you put a cell that likes to attach and to migrate, they become very frustrated in these matrices. So these are cells found in your bone marrow that they're trying to attach to the matrix. They're trying to move towards one another, and they can't. They're trapped inside this covalently cross-linked network, and they can't attach even to the network structure itself to gain traction to try to squeeze through this pore structure. So where that's led us is that oftentimes you're interested in how can I entrap cells or culture cells inside or on hydrogel systems, but then induce some type of dynamic change that allows cells to interact with that material or allows cells to move towards one another. And what I'd like to tell you about are 2 types of systems that we're very interested in. The first one I'm going to tell you about is called user-dictated. Well, I'm sitting as the experimenter watching those cells in real time trying to migrate towards one another in that hydrogel system. And I want to come in with a laser beam or a light source and induce a change and watch how the cell responds to that change. So that's one of the first chemistries. So we're going to put in different types of degradable moieties into our PEG crosslinker. And I'm going to walk you through a little bit of the chemistry of a recent PhD student who just joined the faculty at the University of Delaware. And what she did was April developed these nitrobenzyl ether molecules that are acrylated monomers. And this group here, if you select light of appropriate wavelength, undergoes a cleavage reaction, an intramolecular cleavage reaction, and it's irreversible and it'll break the bond right there. So April made that monomer and attached it to PEG. And so now what happens when you take this crosslinker, you can copolymerize it with other monomers. Now you make crosslink networks. But what's different about this network is when I expose it to light, I get cleavage in the crosslinks. So now I can start to have a dynamically tunable system where I can change the network crosslinking density, which influences many properties. I can erode the gel. I can change its mechanical properties, diffusion. And so let's just characterize this. So here we're using a technique, so it's just rheometry. And so if you put your gel sample on a rheometer, and you have a little attachment here where I can introduce light of a particular wavelength, and now I can measure the elasticity of that gel, and that's directly proportional to the crosslinking density, how many crosslinks are intact. So that's just rubber elasticity that will tell us that. And so now if I follow this in real time while I'm exposing to light, then I watch the change in the elastic modulus as a function of exposure time. And I see this exponential decay in the modulus. And I can fit that, and it'll have a time constant here of about 140 seconds when I use 20 milliwatts per centimeter squared of 365 nanometer light. If I half the light intensity, I approximately double that time constant. And if I change the wavelength and go out to 405, then I also shift the time constant in a manner that exactly correlates to how that molecule absorbs light at that wavelength. And so the nice thing is that my modulus, I know, should exponentially decay with time because my cleavage kinetics, just from photochemistry, I know inversely depends on my light intensity and my molar extinction coefficient at that wavelength. So you have a predictably tunable change in the gel's cross-linking density. So now, what can we do with this? Well, one of the interesting things is now I can have a material that as I expose it, if I shutter my light source, so I can have a material that has an initial elasticity, expose it for 2 minutes, and then remove the light source, and now my material has roughly half of that initial elasticity or stiffness. And then I can re-expose it and soften it again to 1/4 of its original value. And I can step through and change material properties by just the gradient or change in the exposure time profile. So let's use this to answer a biological question. So let's step back to that original motivation that I showed you about those valve cells, that when you cultured them on tissue culture polystyrene, a glassy polymer, that they behave very differently than they do in the soft tissue of our heart valves. So what happens is when I isolate these cells, put them on a plate, within 24 hours, you'll get 90+% of your cells become what's called an activated myofibroblast. And the hallmark of this is this green stain you see here. The blue dots here are the cell nuclei. And the green is a protein that the cell is making called alpha smooth muscle actin. And that tells me these cells are becoming a myofibroblast. Now that's, for those of you that are interested, this change from this fibroblast-like cell to a myofibroblast cell is really important in a lot of wound healing. When you cut yourself with your skin, the fibroblasts in your skin become activated myofibroblasts. They migrate to the wound site, they contract it, they secrete lots of tissue. And then you have regenerated skin, or sometimes a scar. Well, what happens in your heart valves are these thin little tissues that act like perfect one-way valves in your heart. And this thin little tissue, imagine your aortic valve. there's a huge pressure drop across your aortic valve as it directs flow in your heart. It has to open and close 60 to 80 times a minute. And so that tissue is constantly remodeling itself because of all those mechanical stresses. And it does that become activated myofibroblast. But the problem is if this activated myofibroblast persists, these cells keep making more and more tissue. And your tissue becomes fibrotic and scarred, and your heart valves get thicker and thicker. And pretty soon they don't open and close properly. And eventually, if it gets so stiff, the cells will start to express genes found in osteoblasts. And that means they'll start to calcify. So now let's watch over here. Here's what happens if you try to culture these cells in a standard way. Put them on a plate. 24 hours, they become activated myofibroblasts. They contract, they snap, and they start to form these nodules. And they'll even begin to calcify with time. So this type of culture environment is much more reminiscent of a disease state than of a healthy state. So what we were interested in was trying to understand what's the role of substrate elasticity in causing this activation. The stiffer the substrate that's what's causing this activation. And then probably more interesting is what's the fate of this myofibroblast? When we're healthy, the cells activate, and then what happens to them after they activate and maintain the proper tissue structure? So we were interested, what would happen if we could reverse the stiffness of the substrate? Would these cells apoptose, that means die off? Do they go back and just quiesce or go deactivate back to this fibroblast phenotype? And this is something that biologists don't know the answer to, but yet it's really important for understanding a lot of disease. So here's the way we used our materials. We made a slab of our material that we were going to culture cells on. And then we passed a plate over the top of the material at a very defined speed to create a gradient in light exposure. So that gradient in light exposure causes a gradient in the elasticity as a function of position around here. And so these are the experimental points measured with nanoindentation. And then the red is just what we would predict based upon our characterization by the rheological properties, that characterization that we did for the kinetics of cleavage. All right, now let's seed cells on these. So the first thing we had to decide was when I seed cells on these, there's a gradient in the mechanics, the elasticity of the surface, and we had to rule out that the cells wouldn't all migrate to one side or the other in response to that elasticity, because we wanted them to stay in their local environments and sense just that local stiffness around them. So we cultured cells, and we wanted to make sure that they stayed distributed like this. So we did some real-time cell tracking and sampled across here. And these are just tracks of cells in X and Y in this material system. And you see that they take random walks, and the random walks and the velocity crosses 0. So we kind of ruled out that they were moving at most a few hundred microns in any direction, but it was random and largely staying in their local environment. Okay, so now the next question we asked, so if I seed cells on this environment, will there be a change in activation depending upon their position? And so now, I'm going to dim the lights a little bit here. We use that marker, that alpha smooth muscle actin, for the myofibroblast. And so here, blue is the cell nucleus. Red just tells you where the cell's cytoskeleton is. And green is the marker for the alpha smooth muscle actin. So at least in this one, I think you can see more green on the stiffer side of the substrate. And if you quantify all that, you can see that the percent of cells that are myofibroblasts tracks nicely with stiffness across the gel. So on this softer substrate, I get very low percentage of myofibroblasts, and on the stiffer, I get a very high level of activated myofibroblasts. Okay, so that was our first question, but there are lots of different materials that you could use to answer this question. So now what's unique that we can do with these phototunable materials? Well, now we can do the experiment where we culture cells on these stiff substrates that should activate them after 3 days. And then we can ask the question, what happens after they're activated and if I soften that substrate? Again, will they die? Will they apoptose? Will they deactivate? And so here's just confirmation that we culture cells on the stiff substrate, they activate and stay activated over the time course of our experiment. Now we take one of these samples and we soften it at day 3, and after we soften it, we see that the cells, in fact, deactivate. So they don't apoptose, but they undergo a reversal in their phenotype. And they behave as if they were always cultured on the soft substrates. So here's the quantification of that. So we can culture cells on stiff gels, they activate, stay activated. Soft gels, they stay low level of myofibroblasts, and here we can activate, and when we soften it, it reverses. So that's something that we're very interested in. We collaborate with some cardiologists to try to understand how we might be able to use this to regulate fibrosis and myofibroblast populations in people with heart valve disease. Okay, let me turn the light back just a little bit. So another thing about when you use light to change material properties is that you can spatially control where the reactions are occurring. So now another question that we have is we can observe changes that cells have in response to this change in elasticity of their environment, but how and why? How do cells respond to stiffness? And so now we culture cells on these substrates. I think I'll dim this again just for a bit. So we culture a cell on this substrate, and now I can come in with a focused laser light, and I can use a 2-photon confocal microscope to erode the gel in certain regions around the cell. And that removes adhesive context to this cell, and I can watch how it responds and how much it's pulling or tugging on this matrix. So I'll just show you, here's one example where we're going to erode out in this region of the cell, and then in real time we watch how the cell cytoskeleton retracts and remodels. And I think this is probably best seen in this little video here. So when I click on this, that'll be when the degradation occurs. Oops. And you'll see the cell dramatically, so it has a lot of pretension against the material that it's tugging on, and it rapidly reorganizes. And if you look closely, you'll even notice that this cell here responds to this change here. So we're using this to understand dynamics and relate this to different stiffnesses of substrates. So we can model this much like a dampened viscoelastic retractive spring, and the time lag that occurs, as well as the time constant for this remodeling. And when we fit these types of retraction experiments as a function of time, we can learn from this and compare to things like when cells are cultured on tissue culture or glass. And in fact, we see about 6-fold slower responses in these time constants and time lags. And we're using this to try to better understand mechanotransduction principles in cells. Okay, so one other aspect, these were both physical properties of the gel that I was tuning. Its stiffness, eroding regions where the cells are attaching to the material. But sometimes you'd also like to manipulate the biochemical functionality, a signal that a cell receives in this context. And what happens if it's present for a certain amount of time and then I remove that biological ligand? And how does a cell respond to that change in its environment? And so that same type of monomer that we used before, we can attach any amine-containing molecule, like a protein or a peptide. And here's one peptide sequence that's widely studied in the biomaterials community. It's a 3 amino acid sequence of arginine, glycine, and aspartic acid. And it's found in many different adhesion proteins that are found in your tissues, such as fibronectin. And so now I can put this ligand that cells can attach to and then have it be degraded or cleaved at a certain point in time. And so one of the questions that we use this is we were interested in taking these bone marrow-derived stem cells and we wanted to turn them into chondrocytes to use for cartilage repair. And if you look at what happens naturally during development and in our growth plate, as these bone marrow cells are helping to contribute to cartilage growth, is that the first thing that these cells do is that early on they'll upregulate this protein, this fibronectin. And they make that right around their environment and they attach to it and get survival signals. And then they'll begin to initiate signaling associated with their chondrogenesis, or becoming these chondrocyte cells. And after about 10 days, that means they'll deposit one of the main components of their tissues. It's a polysaccharide, a glycosaminoglycan. And they'll downregulate this production of the adhesive protein. So they're starting to build the tissue and remove some of these immature markers for their state. And then later on, this will become more mature as they're beginning to make their own chondrocytic niche. So what happens is that when we culture these MSCs inside of these gels and we give them the RGD signal, we know that it's really important for those cells to survive. So early on, we get very high levels of cell viability when RGD is present. But if we culture the cells in the absence of that signal, about half of them die off after 4 days. So we put that signal in place. But now our question is, a lot of times we'll put that signal in to help cells survive in our culture. But what happens if it persists? So we had the hypothesis that if the signal stays around too long, it will prevent the cells from becoming mature chondrocytes. And so we said, well, what if we could remove that signal at any day and time? And with the photolabile chemistry, I could pick 4 days, 10 days, 14 days. So I'm just going to show you results, what happens at 10 days when you remove it. So that's going to be our experiment. We're going to culture these cells, and then we're going to photolytically remove the RGD. And we're going to look at these chondrogenic markers. And so the first marker that we're going to look at is that glycosaminoglycan, that GAG as a function of time. And when RGD is present and persisting, we get small amounts of the GAG being secreted and made by the cells. When RGD isn't present, it's low, but this is low for a different reason. The cells aren't surviving. And if I photolytically remove the RGD here at day 10, I see a major uptick in the secretion and production of these GAGs. So that's just one marker. We've looked at many different markers. And we also try to observe this directly. So cells bind to this RGD through one of those surface integrins, or ligands, and one of them is an integrin called alpha V beta 3. And if we look at this, so what happens, if I stain for that integrin directly, when RGD is present, almost all my cells are positive for this alpha V beta 3 integrin. And if RGD persists for 3 weeks, almost all of them stay positive for that. But if I remove this signal at day 10 and then analyze for this integrin at 3 weeks, I see a very low number. So this is one piece of evidence that the cells were binding to that RGD and they lose the expression of that integrin at high levels when the RGD signal goes away. And then I can look at different markers. So here's telling me, is it staying as that MSC, so this one called CD105, or is it becoming a more mature chondrocyte which expresses another molecule called collagen II? And now I'm going through this somewhat quickly, but the take-home message is early on, almost all the cells are in their stem cell phenotype, so this MSC marker. And then if I look at after 3 weeks, if my RGD persists, I get some cells becoming chondrocytes or expressing mature chondrocyte markers measured here, but I can enhance that significantly if I just remove one of these signals, up to 75% by just cleaving RGD. So this brings just a new idea or concept that many times in biomaterial design, we look at what signals should I include to promote cells to have certain functions. But it can also be very desirable that the temporal presentation of those signals is really important as you're trying to coax different types of progenitor cells to become specific cell types in our body's tissues. All right, for the last part of the talk, I would like to to now step away from these user-dictated experiments, degradation. But there's also a complementary. Sometimes what I would like to do is I want to be the experimenter and just watch the cells as they naturally would behave in a tissue. Watch and observe them in something that's a tissue mimic, and then maybe later on I would like to trigger or cause a change. And so for this, we're interested in different cell-dictated degradations of the matrix, much like they do in the native tissue. So a cell can decide there's a bond here, it secretes an enzyme or protease that can cleave this, and they decide when they're going to expose a certain signal. So the chemistry, or what we do here, is we look to the natural extracellular matrix in our body, which has all types of complex proteins and sugars in them. But if you boil it down to a very simplistic sense, many of these that are hydrogels that we just worked before, they have high water content and they give mechanical cues. That's what my previous systems did. But the native matrix also has things that sites for cells to adhere to. Specific regions that cells can create proteases to degrade it. It's also a reservoir for storing many of the different growth factors that are important for maintaining our bodies in their homeostatic state and in wound healing as well. So rather than using these full complicated protein molecules, which can be challenging to engineer, we take a simplified approach and we just make peptides as mimics of those proteins. So we're learning a lot from structural biology so that we know, I just gave you an example of RGD is where cells bind to fibronectin. We know different sequences that are found in collagen and different matrix proteins that cells will make enzymes and cause a cleavage right here. We also know different affinity sequences where in the matrix proteins will bind. So here's one that binds a growth factor called transforming growth factor beta. So now what we do is that here's a little bit of chemistry, a second type of chemistry, where we take our same favorite PEG molecule, but now we're going to use a different reaction molecule because I want to incorporate peptides into this PEG molecule. And with peptides, I'm going to use this molecule, cysteine, one of the amino acids that has a thiol. And so now what I'm going to do is, so my thiol is fixed, I'm going to pick very specific end functionalities to my PEG, different ene functionalities that will react with these thiols. And so we've done quite a bit of work by picking enes that, here's what happens. I expose this to light, it induces this thiol radical. That thiol radical will propagate through selected enes, and when it propagates through that ene, it forms this carbon-based radical. Now, interestingly, if I pick my ene appropriately, this carbon-based radical will not propagate through this ene. It can only undergo a chain transfer reaction to the thiol, creates a growing polymer chain, and it recreates this thiol radical. So it's a really interesting reaction because it's a radical-mediated process, but it's a step growth polymerization. And so now what I can do with this system is click together different peptides and PEG molecules in a stepwise fashion. And so now I'm going to use the same rheometer and now measure the polymerization, the gel evolution instead of degradation. And so you can see we've studied a wide range of different enes. And this cyclic norbornene is one we use a lot because the kinetics for the chain transfer reaction is very fast. And so we get complete gel formation in 2 or 3 minutes. And this point here is roughly where the elastic and viscous modulus cross over. And so that's an estimate of when it forms a gel. So you form a solid gel in just under 15 seconds. So now the idea is, with this reaction, it's quite simple. You take PEG molecules of different molecular weights, different number of arms on the PEG. You take peptides of various sequences or interests, and so you tune physical properties with your PEG, biological signals with your peptide, and now you can begin to screen and try to understand cell responses of interest. And so one that can be of interest is just how motile cells are in this matrix. And so if we click together gels that I'm going to use the RGD sequence for adhesion, and we'll use this degradable sequence where I'm going to make a point variation and replace this tryptophan with an alanine. And let's watch these cells migrate through this. So here, the tryptophan is highly susceptible to degradation by lots of enzymes the cells secrete. And the cells now migrate through this as compared to those cells that were trapped inside of those matrices. And if I just do a small variation to this tryptophan, replace it with an alanine, it slows down degradation. So cells can attach to this matrix, but they can't degrade it as quickly. And so their mobility is greatly diminished. I put up one image here, is that one of the tricky things, so we can also characterize how cells interact with this type of material, and then try to relate it back to their structure and function in a native tissue. And that's one of the big challenges, of trying to better understand how in vitro culture conditions can match this in vivo phenotype. But I'm just going to show you one simple result that we did, where this image here is a critical size bone defect in a rat's skull. So this is a CT image. So what that means is that a hole was drilled in the skull of the rat right here, and it was of sufficient size that it won't heal by itself. Now, if you put in two of those PEG formulations, and so this says non-degradable, think of that as that alanine variation where cells couldn't migrate through it very well. And here's that tryptophan version where cells could degrade at high rates. So what's interesting is just by putting in a matrix that cells can migrate through, you can promote a significant amount of healing in this critical size defect. So there's no drug, there's no growth factor, it's just a material environment that provides an environment for cells to migrate through. So one, that was really interesting to us. It was kind of an unexpected result. But now the engineer in us has said, well, so that was pretty exciting, but it's still not completely healed. And if I showed you all the details, you'd say, the bone, that's pretty interesting bone regeneration, but it's not exactly the same as the native tissue. And it still takes about 8 or 9 weeks. So then now, how do you make it better? Is it that I need to have a system that cells can migrate through faster? Is it that cells need to migrate through and encounter some signal that tells them to secrete and make mineralized tissue faster and change their secretory properties? Or does it need to get some signal to tell them to increase in their number and proliferate? So this is where I think some of the combinations of chemistries can become very interesting. And doing these types of thought experiments where now I can watch cells in this nature of environment and then on demand with light deliver different types of signals and see how those cells respond. So I'm just going to give you one example of how we do sequential and orthogonal types of reactions, and I'll go through this in the last 5 minutes. So the chemistry is going to be very similar to that photodegradable system, but all we did now is we applied some polymer processing techniques where we did a microsuspension polymerization and made small microparticles out of these. And we made these microparticles so we could load them up with different types of proteins or factors of interest. And we can characterize this distribution of the particles. And we wanted to keep them big enough so that they wouldn't get internalized by the cell. They'd stay external, but not, and we also wanted to have them big enough so we could image them, but not too big so that when we released the load, we'd have too high of a dose of the protein of interest. And so we loaded up these particles with different types of proteins, and here's just some fluorescently labeled ones. And now, I'll just show you one little movie here where I have these particles loaded up with a fluorescently labeled protein, and now I'm just going to expose it to light, and you can watch. As you expose it to light, it swells a little bit, and then in about 40 seconds, you have complete erosion of the particle, and it releases all of its contents. So you can characterize this, and we like to quantify and understand. So we look at what happens to the volume of the particle, and we can understand what happens to the crosslinks in the particle as it swells and increases in size from thermodynamics. And we can follow the fractional release of the protein with time. And as you degrade the system, you get some swelling that causes a small fractional release of the protein. And then here your particle completely dissolves and is eroded, and you release the rest of your payload. All right, so then what this can be useful for, so that was just a model protein, but you care really about whether proteins are active or not. And so that was the next thing that we did, was we put in a growth factor called transforming growth factor beta. And we used a cell line, it's just a reporter cell line. So it's, they're called PE25s, and they have a reporter so that when TGF-beta is released to the cells, it causes them to make luciferase, which then I can detect. All right, so I seed cells on these with these particles, and then I'm going to expose it to light. And so here are the basal level of the cells in media and their luciferase production. Here's blank particles with no TGF-beta. Here's what happens when I release them, and here's what happens if I were to just add TGF-beta to the solution. So we have a significant signal from the controls, but it's not quite up to what it would be with soluble TGF-beta. So proteins, it's really difficult when you encapsulate them because you can denature them very easily. So we're working on techniques to improve the activity of the protein, but we can release and get active protein release that can influence cell signaling. So now I'll just tell you where we're thinking and going with this. So now we're going back to this experiment where we had this untreated control, and then we had this one particular formulation that cells could degrade through and cause bone regeneration. Well, in the past, what you would have to do is you would make microsphere formulations, and you might deliver a protein of interest, like a bone morphogenic protein, and you'd be interested in, well, how should I release that to accelerate bone healing? It's used as a commercial product. And here's what happens if you take the commercial product dose. So some of you may know this from the recent press on this, in that BMPs are a very potent molecule. And right now it's used to help bones heal faster in spinal fusions. And in fact, if I do a quick head count of the people in this room, if I took all of the BMPs in your bone marrow, I could treat one person. So we aren't delivering it efficiently. And if you take that dose and you put it into this polymer, what happens is you get super physiologic bone growth. And so you get all these side reactions, bone that will form on the top of the skull and some in the material. And this product got some bad press because it was used for spinal fusions in small infants that had congenital defects, and it caused a side reaction that caused their airways to swell, and they had to remove this BMP. So they have to be really careful of what they use it for. So we're interested, classically, you'd have to formulate microspheres, characterize what their release profile was, then put it inside of this bone defect and say, did I get it right? And if not, then you'd go back. And so here was one of our guesses, where in about 4 weeks, we did pretty good to what we saw up here. It was faster. But now what we're doing is we're putting in photodegradable microparticles into these bone defects. And now I can put a little window on the top of this rat skull and watch healing occur and release those particles and see what is the optimal profile for this type of release, and learn something fundamentally about whether this is the right target or whether I need to have more cells infiltrate. So what I hope I've shared with you today is that we've had a lot of interest in developing different types of photochemistries that can be done and reacted, polymerizations, in the presence of cells. And this provides a really versatile platform for the three-dimensional culture of cells. I showed you examples where we tried to use these dynamic environments to answer really fundamental biological questions. How do VICs become myofibroblasts and deactivate? How do these MSCs differentiate into chondrocytes and the role of temporal signals? I think there's also a lot of power in using orthogonal types of chemistries, so the thiolene chemistry, where I can watch and encapsulate cells and observe them, but then my independent photochemistry with the particles, where I can release signals within that environment at a different point in time. And I think this is where we'll use to circle back to try to better develop procedures for applications for regrowing and regenerating tissue structures. So with that, I'd just like to acknowledge a great group of students and postdocs that I've had at the University of Colorado. Everything I showed you today, unfortunately, I didn't do any of the experiments, and I'm not very good at doing experiments anymore. But they're a great group, and some of the people whose work I told you about, the photodegradable particle was started by Andrea Costco and April Clarkson. And then Chelsea helped with the chondrogenic work with the MSCs, and Sharon worked with the VIC work with the myofibroblasts. And then all the photodegradable microparticle works was Mark Tibbetts, and Pete Mariner, Kyle Carverts, and a collaborator, Chris Bowman. We did a lot of work with the thiolene chemistries to make the peptide functionalized pegs. And with that, I'd be really happy to answer questions that you might have. Yeah, I think that so it's not something that we've focused on, but I do think that there are electrochemical gradients and ways that you can move cells around with that as well. Yeah. Okay.
Is there a concern using like high energy light in terms of like DNA?
Yeah. So the question is about the wavelength of light inducing cell damage. And so yes, I mean, so 365 is actually really low energy ultraviolet light, but it's a common question that we get. And we've really truly looked for DNA damage at the doses that we give and can't see it, but that doesn't mean it isn't occurring, and cells have a lot of repair mechanisms for that damage. But all the chemistries that I've showed you are completely compatible with different wavelengths if you choose different initiators to go longer wavelengths. The nitrobenzyl ether group, we can use 405 and longer, or if we use 2 photon, you can go out to 720, 730, depending on lasers that you have. But yeah, it is a concern, something to watch for, especially for the translation of these applications to in vivo work. So the question is about the distribution of the cells. And so that's something that we can control very easily when we do the encapsulation. And you can look up average density of cells in many of the tissues. It might be about 10 million cells in a cubic centimeter, might be a typical density. But what we haven't done is we haven't tried to organize the cells as they might be in a tissue. So we can homogeneously control that distribution. Oftentimes in certain tissues, and as you go to larger size scales, you'll have cells organized in columnar regions or in different horizontal flat regions across a tissue. And that's something we've explored a little bit, but none of these strategies would organize the cells in that fashion.
I have a quick question for you. So when your cells, particularly the MSC to chondrocytes, and you're adding the RGD, I was a little surprised to see they're still very round. Is that a physical constraint of the hydrogel, or is that you don't have a lot of RGD? Because I would have guessed that having that adhesion, they would spread out.
Yeah, no, that's a good question. So the work I showed with our MSCs, that gel was non-degradable system, so it forced the cells into the round morphology and just interacting with RGD. If you did the same experiment in the thiolene gels, you would see the cells much more spread and attached.
So what I'm wondering is the materials that you make when you make each one of these, change the chemical, forgive me, I don't recognize it, but is the end result the same? You showed the bone growth in the top of the rat's head, and you said you showed how it grew more when you used one of your techniques. Is the material the same?
Yeah, so what was different in that, so we used the exact same material, and then what we did was we used, in the one where we got super physiological bone growth, we put a protein in it, the BMP, that's supposed to promote bone growth. And if you just entrapped it at a particular dose that's used in the commercial device, you got all types of what we call superphysiologic bone growth, almost like a big cap that would form them. If you put these in microparticles where you tune that release and you went to a lower dose, then we could start to get faster bone regeneration and in a defined volume. But part of what we're struggling with that approach is that classic microparticle techniques, you have to go back and reformulate, and is that right? Go back and reformulate, and do I have this right? And so where we're going is we're using the photodegradable microparticles in the system. So now you just load up your particles at one particular formulation, and you use light to change your dose at any instant in time. So now you just have a single formulation, and you can test many different conditions.
So, Kristi, in regard to phototuning materials for manipulating mechanical properties, is there an issue with temporal stability of these materials?
Yeah, so I'm not sure where we've done any long-term exposures, but so these samples can sit out in room light for several weeks without a change. So it's both the intensity of the light, so it's a dose, right? The intensity over the time course, I would say. But yes, if you wanted to store them for 2 months, then you might have to worry about stray light causing some instabilities or degradation prematurely. Yeah.
Okay, so my limited knowledge of biology tells me that usually organisms will use multiple signaling mechanisms to control the way structures will behave. So when you're trying to create truly physiologically equivalent structures and you have only a finite number of knobs that you can tune, how close do you really think you can get?
Yeah, I don't think we recapitulate everything that goes on in the in vivo environment. I think some of the first part of my talk was sort of like, okay, let's try to pick this apart and look at signal by signal. I think where we're going with some of the things is, now we have more tools to look more globally at how cells are responding to changes. And so what we know are things like cells on tissue culture plastic, so they might express one gene, they're over here. And cells in vivo might be over here. And so what we're hoping is, as we have better tissue-like environments, is that maybe we can get more of an overlap that's like this. But I don't think we're going to purely in an in vitro sense recapitulate everything, because there's a context of surrounding cells and tissues and secreted hormones that act over longer distances. And, So right now we think more of the applications would be in vitro. So one example is in all the stem cell cultures. So now you could envision a lot of stem cells are even grown in aggregated structures. If you have particles within them, now you can more uniformly release factors at appropriate times. I think you can do particular thought experiments, like with the bone healing model, that I think that's going to be a really rapid way for us to figure out what release profile that we would like for BMP. And then once we know that profile, then we can formulate the system to release at that profile, and that's the one we would use for like a clinical application.
So with these microparticles that you're using laser light to control their release,
Yeah.
How far into skin or biological material does that light penetrate, and what kind of release mechanism would you use if the light doesn't penetrate deep enough for some of the applications?
Yeah, so all of what we'd done to date was in vitro culture methods. I had a former fellow who's now in dermatology, so we're looking at some dermatologic applications for wound healing. There you can go reasonably deep through a millimeter sample of tissue. But I think, yeah, if you were looking at something that was much deeper in the body, you'd probably have to change the chemistry of the degradable group, so it might be more sensitive to something like near infrared or something, a different wavelength that would penetrate more deeply.
If not, Kristi, thank you from the school. A small token of our gratitude. And please join me in thanking Kristi once again.
Thank you.